really excited to introduce Toby. Dr. Tobias Beyer is a Chief Scientific Officer and Lead of Business Intelligence at Cytosurge. He has over 20 years of research experience, a decade of R&D leadership, and he specializes in cell line engineering, stem cell biology, and CRISPR-Cas9. After earning his PhD at ETH Zurich and completing a postdoc at the Bloomfield Tenenbaum Research Institute in Toronto, he served as a junior group leader at ETH Zurich. Today, Toby bridges science and business, leading cross-functional teams to develop applications for the FluidFM OMNIUM and driving innovative ideas from concept to commercial products. So I'm excited to learn a bit more. Thank you very much.
Thank you so much, Harry. Thank you so much for being here. As Tarek mentioned, I'm the Chief Scientific Officer of Cytosurge, and what I'm trying to do is find people that are interested in using our unique technology. So what you see here is basically our device. It's an inverted microscope that also has an incubator on top of it, because people always ask me, oh yeah, do you have temperature and CO2 control? And yes, we do. And now, aside from the instrument, I would like to switch over to the more scientific part. And usually I start off with like an experiment, the process of epithelial to mesenchymal transition, a very dynamic process. You have the epithelial cells, you have the hybrid state, and they become more and more mesenchymal. But it's not one direction they go back and forth. And this is a real hard process to study. So what is being done mainly now on the transcriptome side is that people use single-cell RNA sequencing, like 10x genomics. They take snapshots at different time points, and they kind of chart out the whole landscape. So it's really great to provide the cellular sensors. You know what's there, but you have one major drawback. And this is, if you want to measure the cell, you have to destroy it. And that kind of gives you the ability to directly follow a single cell, where they see what it will do, where is it going, and you have to rely on inference and correlation instead of causality. And this is where our device comes in. We can go to one single cell, we can poke it, we take out cytoplasmic biopsy, we call it, we can record the transcriptome, and the clue on the whole thing is the cell will stay alive, so you can follow it, you can treat it, you can even go and poke it again and measure it again. So how do we do that? We have a crossover between atomic force microscopy and microfluidics. The core is this nanosyringe, so more or less the smallest syringe in the world. So the opening here you can see is about 600 nanometers, which is about 10 times smaller than your regular mosquito. The other thing is the cantilever of the FluidFM is hollow, and we mount the whole thing on an inverted microscope. So you have your cells here. This is the cantilever, and like in the regular AFM, you have a laser beam that deflects from the cantilever. If the cantilever touches the surface, it starts bending, and you can calculate back afterwards the force you apply to it. And with this, we can very, very gently interact with single cells. And then we couple the whole thing to a microfluidic control system, and then by either applying positive or negative pressure, we can inject or extract from a single cell without destroying it. Just to sum that up, so we have physical access to cells. We are very, very gentle because of this force feedback control. We can go into the sub-picoliter range, and the geometry you see here can be adjusted to either fit different cell types or cell heights. How does that look like in the real life? So we have the workflow here. You have, again, our epithelial cells. You poke a couple of them, set the sample you've taken aside, freeze it down. You stimulate the cell, you start following it with live cell imaging, and after 4 and up to maybe 36 hours, you can go and take another sample. What are the readouts? So we can do the transcriptome. We can look at the proteome. And on top, because you have an inverted microscope on it, you can record how the cell looked like. So you have the morphology. In the case you have a fluorescent marker in there, you can also do fluorescent imaging. And last but not least, you can also record the context. So if you have two cell types that interact, you can kind of go and correlate the distance between a macrophage and your cancer cell and see what kind of effect that will have on the transcriptome. So briefly, what you have is, instead of just 2 dimensions in your UMAP, you add a time dimension and the phenotypic dimension, and this really gives you deeper insight into what a cell is doing. So this was very theoretical. So what I like to do now is I will just show you how that looks like in real life. So this is the interface of our microscope. So the operator marked the GFP positive HeLa cell, initiated the process, so all the parameters are set. The cantilever is approaching, piercing the cell. Now we apply negative pressure. And you can see a bit how the cytoplasm comes into the cantilever. And to illustrate it better, we go now to an empty valve where we don't have background fluorescence. And what you can see here now is the cytoplasma that we just took out. And this is because we know the dimension, we can calculate back afterwards how many femtoliters in this case we took out. So what are the use cases? So first I will just show you a couple of things that are already published. So we have a scenario where we have macrophages. They carry an LPS and a kappa B response element that drives M-cherry expression. The cells can be stimulated with LPS. And you all know these cells are, in theory, they're the same, but they will react differently to a stimulus. This is exactly the case when you do the live cell imaging. You can see one cell has an extremely high response, another one a bit less, and some of them don't react at all. Of course, you can go now into single-cell RNA-seq, which is great, so you can see the different gene expression at time 0 and time 4 hours. And in this case, when you look at the data, you see that NF-kappa BIA positively correlates with the TNF expression. So you would say, okay, they kind of act together, and that's how that pathway works. However, if you do it backwards, you take the transcriptome first. So you take a biopsy with our device, you stimulate the cell and follow it, and then integrate the data. Instead of this positive correlation, what you see is exactly inverse. In this case, cells that respond very highly to the LPS response have very little NF-kappa BI at the beginning. And this is kind of a paradoxon, but if you start thinking about how the pathway works, it makes absolute sense. So looking at the NF-kappa B pathway, you have NF-kappa BI as an inhibitor that binds NF-kappa B. Upon the stimulus, it gets released, it goes into the nucleus, and it binds to TNF-alpha, but also on the NF-kappa BI promoter, so you get a feedback loop. And when you look at just endpoint data, you would kind of draw the wrong conclusion that when you have loads of NF-kappa BI at the beginning, you would get a high response. But because you get it the other way around, this is exactly the things you can discover using Live-seq. Next application is looking at cell fate trajectories. In this scenario, in that paper, they poked the adipocyte precursor cells. They differentiated them into adipocytes. And here they combined single-cell RNA-seq with the live sequencing. And basically what you get is, because we know, or we measured it, we get the ground truth. We know exactly from where the cells are and where they're going. And you can say, okay, you can do that with inference methods, but basically that's what you get when you do inference. You get the pathway of the trajectory the other way around. You get weird things, so you don't really see it at that resolution. So what we can really do is, because we have the ground truth, because we know the beginning and the end, we know exactly how these cells move through their fate. Another example, so this is done together with a client of us. So how we operate usually when someone would like to have a device in their institute, they come to us with a minimal experiment. We do it in our facility. We provide them the data so they can go and apply for a grant to obtain the device. And what we did is, in this case, is we have cells. We stimulated them with conditioned media. So we took T0 time point, T1 time point, and we did the transcriptomes. And this is just basically the controls and how the gene counts and the total RNA counts look like. So the negative controls, what we usually do is we pipette the buffer back and forth to make sure that we don't have any carryover. We have positive controls, so this is 1.0 0.1 picogram of total RNA, 10 picograms of total RNA. The biopsies are somewhere in that range here. And these are two independent replicates, so they are kind of comparable. And if you start looking now, okay, what happens to those cells? You see something very interesting. So when you look at PCAs, you have two cell types. So these are the untreated cells are in blue. The treated cells are here, and you can see kind of they bifurcate and go two different directions. And here the same thing again. And if you look at the heat maps, this is exactly what we observe. So what we can see is that depending on their pre-transcriptome, these cells take different routes in when they are stimulated. And this is also something that you cannot really do with single-cell RNA-seq. Usually, the first question we get is, okay, what's the throughput? And yes, throughput is not so high, but I just want to show you now that what we did with this initial paper from a collaborator, we started automating it and bringing it up to speed. So we go back to this first figure here. So they used the so-called add-on on an AFM. So you have a very small view of field. You don't have a lot of space where you can operate. You can do maybe 10, 20 biopsies before a day. The library prep was done by a homebrew SmartSeq, enhanced SmartSeq 2, and about 60% of the libraries failed because it was so little of RNA. Illumina-based sequencing, and we estimate that the postdoc that did that experiment was collecting about for 6 months data points. And it's also highly dependent on the operator skill. So what we did now is we took the workflow, but we adapted it to our newer version. And with that instrumentation, you can basically go on the whole field of a 6-centimeter dish. It's semi-automated cell tracking, and we can go up to 40 biopsies a day. The library prep, we adapted the commercially available library prep kit to make it easier for clients. That bumped it up to about 60-70% success rate. The sequencing stayed the same, and we cut down the data collection to about 1 to 2 months. So that's almost a 5x increase. And it's also not dependent on the operator. So we have a whole lab of people that are doing these experiments. So once you get an introduction, you can do it on your own. So one of the improvements is how we track the cells. So we have that integrated in our device in the software. If you choose the cell, it starts somewhere here. Yeah, so the first point, and it goes up, and you can follow the cell over 24, 48 hours, depending on how dense your cells are. And so we know exactly which cell we've biopsied so that we can make sure that we go back to the same cell. There's one caveat, and this is just going to happen now. So what are you going to do if the cell divides? Then you have to take kind of the decision which one you're going to poke. But I think this is kind of a luxury problem that the cell is even dividing after it has been taken or biopsied. So how does that integrate or compare to regular single-cell RNA sequencing? As I mentioned in that experiment here, we had both data points. So what we have here is the Live-seq data points, so about 40 and 60, pre and after. So you can get already a separation. You can do the same thing with single-cell RNA-seq. You also get the separation, but if you put them together, because you have more numbers, of course, you get a clear separation. And what's really important here in this figure is when you look at how the cells were sequenced, they kind of cluster not together whether they were Live-seq cells or single-cell RNA-seq cells, but they kind of separate still in their cell fate. Saying that is that we concluded, okay, they are comparable and we can integrate it. Then the other thing is the Luthor HD library preparation kit. This is also something that is a bit particular. So we use this kit from an Austrian company. And the idea there is before you do the cDNA, you amplify linearly the RNA, and you try to avoid to have dropouts. And how they do that, they have oligo T primer with a T7 promoter, and you do the double-stranded RNA, and then you amplify it using the T7 RNA polymerase. Then you have 40 uRNA copies that can be afterwards normally amplified, and then you can do the sequencing. So we did a benchmarking of the kit. So first what we did is we took total RNA from lysed cells. So we had 10 picograms down to 0.15 picogram. So 0.15 picogram is roughly a 200th of a cell. And you can see here at the sequencing depth of 1 million reads, you can still detect about 2,000 genes. So I think this is quite solid. And at a 2/5 of a cell, so these 10 picograms, you're already at saturation, I would say. When you do biopsies, you get a bit of a broader range because you have a sampling bias, but still you get kind of a quite robust readout between 1,000 and 7,000 genes per sample. And this is already enough that you can really learn a lot about these cells. The next argument that most of you probably already have in mind, so, okay, you poke this cell, you take maybe 10 or 20% of the cytoplasma out. So what's the effect of it? That must do something to that poor little cell. And to address that, what we did is we had fibroblasts that were poked, then let to recover for 1 or 4 hours, and then just regular single-cell RNA-seq. And then if you do a UMAP projection, so the red points are untreated cells, green 1 hour, blue 4 hour, and you see there is no kind of clear separation between the samples. However, if you look at differentially expressed genes and you relax the cutoffs quite a bit, you see that there are some genes that are differentially expressed. But what you also see is that you have certain genes that get downregulated in 1 hour, and then they start coming back up. So these are most likely genes that are connected to the cell cycle, because losing 20% of the volume forces the cell to kind of reset and then start growing again until it divides. And then you have some genes that are related to stress response, but you can clearly see after 4 hours, they're already going down. For the cell cycle, so we looked at only the cell cycle genes. So what you can see here is this distribution here. Up here is probably dividing cells. It's a bit hard to see. And down, 1 hour, they are more or less gone, and then they come back up again. And also when you look at the overall viability of these cells, so regular cell lines, these are between 85 and 95%. So most of the cells we find again after 24 hours. If you go towards a bit more finicky cells, like iPS primary cells or iPS-derived cell lines, you see a bit impairment on the viability, but still it's like 70% or more, which is for most applications acceptable. I have to be honest, there are certain limitations that we face at the moment. We do only 2D cell cultures. This is because of the geometry of the device, so we cannot really poke or go into a spheroid or an organoid. And if the cell has a height of less than 1 micrometer, it also becomes difficult because where you position the opening. So in this case, the opening is kind of above the cell, and then you just sample the medium. And if it's higher than 15 micrometers, it becomes hard to kind of penetrate and get in there. And for suspension cells, they are possible, but you have to make them adhere a bit. I have to do a soccer reference here. You can imagine if you have a soccer ball, you press down on it, it will fly away. So you have to make sure that it holds somehow down. So now a bit away from the RNA, this is something that we started about a year ago. We were wondering, okay, is the instrumentation of the mass spec stay sensitive enough that you can measure proteins from a biopsy? And we paired up with a lab in Denmark, and we started doing that. So we sampled again from a cell. We dispensed the cytoplasma into a buffer droplet containing mild detergent and dispensed trypsin in it. So this is about 0.5 microliter to a microliter droplet. Let it digest, and then we shoot it into orbitrapistral. And this was the very first try without any optimization, and we got like average a couple of hundred proteins. And there were, of course, a couple of dropouts. So that was, happens when you do something for the first time. But if you look at the most expressed genes, you can see that they are quite reproducible, that they stay, that you kind of sample the same thing again. We are up to 600 proteins now, and I hope that our collaborator will push out the preprint end of August, and share the workflow, make it available to everybody. Another neat thing is we can also sample not only from the cytoplasm, but also from the nucleus. And if you do proteomics from there and see what you get, you can clearly separate the two compartments. So until now, I focused really on the extraction part, but there is a bit more to come. So we can tailor or change the tips. So this is the nanosyringe that we use for the extraction. We can also use it for injection. Then we have a capped tip. So you can do nanoprinting. So for example, you want to print some attractants or proteins on your dish, you can do that. Then we have the cup-shaped opening for isolation and adhesion force measurements. And last but not least, the special tip that you can use to extract organelles for downstream analysis. I haven't had it here, but we also have a cylindrical probe that is even better for this organelle extraction. I didn't put it here because we haven't commercialized it yet, but end of the year, we should have that. Injections, what you can do is you can go and deliver plasmids, you can deliver linearized double-stranded DNA, single-stranded DNA, RNA, proteins. We did a lot of genome engineering that way. I will go a bit more into detail afterwards. You can also just deliver other proteins or chemical compounds. You can go and isolate a specific cell from a mixture or measure adhesion forces. So to illustrate the injection process, a little video again. So the injection process is a lot more automated. So you can do 300-400 cells in a morning. So you can see here, we had on the right-hand side the parameters. The operator marked 3 cells. The cantilever went to the first cell. You see it's getting marked. And you see pressure and force. And now we will inject a third cell. And note that we have a non-injected cell here and here. What we delivered was Cas9 RNPs that are ATA550 positive. So we switch now to the red fluorescent channel. What you can see here, the slight hue of the injected cell. So we delivered about 10,000 molecules of Cas9 here. You might ask, okay, this is a very complicated way to do genome engineering. And yes, I can skip that. So I don't think I have to explain that. But so why are we doing it like that? So one problem with difficult to transfect cells is that you have to bring the RNP to the nucleus. So it has to pass the cell membrane, face random transport into the cytoplasm, and then go to the nucleus and find the target. And what we do is we just deliver it right into the nucleus where it's needed. An example of what you can do with it, we can do multiplex editing. So we injected a mixture of 3 different RNPs into single cells. You can see here the injected cell. After 3 days, you have already colony. 12th day, you have the full a cell line. About 90% of the cells survived after 24 hours, and 50% formed a colony. And interestingly enough, of those 50%, almost 30-35% had a triple knockout. So you don't have a lot of cell lines, but cell lines you get is what you want. And by adding just a DNA repair template, for example, you can do GFP tagging on tubulin here, or you can do GFP tagging on a nuclear protein. And depending on the cell line, we get about 2 to 7% knock-in rates. You could also go into smaller, like basics changes, also possible. And we busted it up now to a 2.5-3 kilobases into a safe harbor locus. So in the future, we can see that this can be used to deliver constructs up to 10 kb. We provide that as a service. We discuss with the client what they want to do. We design the experiment. We pick up the cells, do the CRISPR design. Then we inject about 300 to 400 cells, single cell seed them, grow them up. And then at the end, you have 2 edited cell lines and control lines. And why? So it's vector-free. They're monoclonal. We can really control how much RNP we put into a cell, thereby really reducing the off-target effect. So you can imagine it's a kinetic experiment. If you have 1,000-fold more protein, the likeliness that some off-target will happen is just higher. Now, quickly, mechanobiology. This is also something that you can do in isolations. So For example, you have one cell in your cell culture that you really want because you want to make a cell line out of it, you want to analyze it further. If it is adherent, we fill the cantilever with trypsin, we shower the cell with trypsin, it starts detaching. So we go down, press a bit on it, negative pressure, and then we can go and put it in your valve. So this is the illustration, or you can do the whole thing with non-adherent cells. So these cells were marked, the cantilever picks it up. We started also automating it that it can recognize a floating cell and go and picking it up. And you just place it in another valve. And at the end, you have these 3 cells sitting next to each other. So it's not the sorter, it is really a picker. Suspension cell, about 60 an hour. Adherent cell, maybe 20, 30 cells. So this becomes really important if you have one very rare or special cell type that you really want to get. Then adhesion force measurements. So you can do that with a regular AFM as well, but there you have to go and glue the cell to your cantilever, you press down, lift it up. If you do 4 or 5 cells a day, you're really good. With our cup-shaped opening, we can go down to the cell, touch, apply negative pressure, and just start lifting the cantilever, and we measure the force that we pull with until the cell detaches. You see now it lifts up, and you see the red line there kind of shows that we reached the rip-off point. We can do a couple of hundreds of cells, so we can really do statistics, and we can kind of characterize the adhesion forces of these cells. Now to the latest or last application, we're just kind of rolling that out now. We have a couple of customers that are already trying to do that. It has been published by one of our partners. So what they did is they played around with the openings of the cantilever, and with a small opening, you mainly get the cytoplasm. If you have a bit bigger opening, you get ER and cytoplasm. So they had ER marked with a fluorescent protein, or you can get mitochondria. And the cool thing what they did is they not only isolated these mitochondria, but they took them and transplanted them into other cells. And you can imagine that this, in the context of aging or cancer biology, this could be kind of a really nice tool to characterize the functions of a really nice functioning mitochondria or one that is just already damaged. This was already published with senescence in CAF-associated, cancer-associated fibroblasts. I already mentioned that when we try to engage with new or potential future kinds, that we offer so-called proof of concept experiments. Let's say you have a drug that works on the cancer cell line. You want to really see how that works on your cells. What we do is we bring your cancer cells to our facility. We discuss how we set up the experiment. We do roughly 20 to 40 biopsies for you. We sequence it and then send the data back. There's also like a rent-to-buy or leasing options, or just come and buy one. Cell line engineering, we offer that as a service. And also we're thinking about having the Live-seq or the sampling as a service. We're building up now our lab capacities, but I think that will take at least until mid-27 until we can really roll it out. Just briefly about us. So we're about 40 employees. We've been founded in 2009 as a spin-off from ETH, so we've been around for quite a while already. There are quite a few publications out there, 140 installations. 20 of them are the newer devices, 4 or 5 in the US already, quite a few in China. Interestingly enough, we have one in Beirut and one in Saudi Arabia. Of course, I would like to thank you for your attention, and please, you can start asking questions.